Good morning, students. This is your teacher Neela Mehlawat, and today we'll be talking about the topic multiple alleles and pleiotropy. So I hope you're ready. So bring your notebooks, books, and pen, pencil. So can we get start? So we'll start with this topic, that is multiple alleles and pleiotropy. Okay. Now coming to the what are multiple alleles? We need to understand what are it. See, generally we know that one gene controls one character, isn't it? One gene controls one character. This was told by Mendel that one gene controls one character. Okay, so we can say normally we have two alleles of a gene. There are two members in a gene, right? But if you have more than two members in a gene, okay, suppose you have a gene and that gene has more than two members, more than two alleles, like A, B, C, then we call these alleles as multiple alleles. We call these alleles as multiple alleles, right? Okay. So we can define now that multiple alleles means when more than two forms of a different gene of a given gene exist in a species. Multiple alleles means एक ऐसी जीन जिसके दो से ज़्यादा members हैं, ठीक है, उसे हम multiple allele कहेंगे. Generally, there is two members in a gene. Now, Mendel has assumed only two alleles of a trait. He said there is only two members of a gene, but in population we find that there are more than two alleles. Okay, and this phenomena is called multiple allelism. Right? Now these we should need to understand the features of multiple alleles. They always present at the same locus. See, we have a chromosome, isn't it? So they will occupy the same position on the chromosome. Like this is the chromosome. So all of them, they will occupy the same position on the chromosome. So they occupy the same locus. Secondly, they control the same character. Means, suppose if height is there, all the three or all the four will control the same, that is the height. If eye of the color is there, they will control the eye of the color. Okay, so they control the same character. And the third, very important, there is no crossing over between these alleles. Okay, so they occupy the same locus, they influence the same character, and there is no crossing over between the multiple alleles. Let's talk about the examples. The very important example is the ABO blood group, right? This is one of the most important example, and this example we'll discuss also. Second is the coat color in the mice. There is a coat color in rabbit and the eye color in Drosophila. These four examples, they are of multiple alleles. Okay, we'll only discuss this example, that is the blood group. Now we know that blood group has three alleles, right? Blood group has three alleles, that is IAB, IB and I. This I stands for the immunoglobulin. Okay, this I stands for immunoglobulin. Let's study the example. I hope you remember this slide. So we have this blood groups and there are ABO blood group system that we are following. Okay, so this ABO blood group is controlled by a gene I. So gene is one, that is I gene. It has three alleles. Let's talk about those three alleles. That is, one is IA, one is IB, and third is I. So here, one gene has three alleles. Okay, one gene has three alleles. And these three alleles, they control the blood group system, right? So three alleles, these are together called as multiple alleles. These are together called as multiple alleles, right? They are together called as multiple alleles. Now if you look at this, IA is dominant over I, IB is also dominant over I, but IA and IB, they are co-dominant to each other. They are co-dominant to each other, right? Okay, let's come to the next topic, that is, this is another example which is there in the NCERT. Let's discuss this example also. Pleiotropy. Okay, pleiotropy means when a single gene produces more than one effect. See, Mendel said one gene will control one character. Okay, this was what Mendel said. Okay, one gene will control one character. Pleiotropy means when one gene will control more than one character. Okay, pleiotropy means when one gene will control let's see this. प्लियोट्रोपी का मतलब होता है जब एक जीन एक से ज्यादा कैरेक्टर को कंट्रोल करती है, मोर देन वन, राइट? So Mendel said one gene will control one character, but pleiotropy, the phenomena of pleiotropy says that one gene is controlling more than one character. Let's study this here. See, this is what Mendel said. One gene will control one character and will have only one effect. Pleiotropy says one gene will produce more than one effect, that it can have more than one phenotypic effect. Phenotypic effect means the effect that we can easily observe. Okay, so we'll talk about an example, that is starch synthesis in pea plants. We'll see how this character is being controlled. The starch synthesis in pea plants. Let's talk about the starch synthesis in pea plants. See, starch synthesis is being controlled by a gene B. Okay, gene B controls the starch synthesis. Now this B gene can have two alleles. One is capital B, another is small b. Okay, this can have two genes, two alleles. The gene can have two alleles, capital B and small b. Okay, so there are three possibilities. One is capital B, second is capital B, and third is small b, small b. This we have studied in Mendel, that there could be three possibilities. Tall also had three possibilities, so this B can also have three possibilities. BB, capital B, and small b, small b. So this is your homozygous condition, homozygous dominant. This is homozygous recessive, and this is your heterozygous condition. When these two alleles, they are in homozygous, capital homozygous, then the seeds produced are round. किस तरीके की seeds हैं? Round seeds हैं. And the starch granules, they are of large size. दोनों features देखिए. Seeds are round and the starch grains, they are of large size. Okay. Small b, the starch grains are of small size and they have wrinkled seeds. Right? They have wrinkled seeds. But when the gene is in heterozygous condition, see, the seeds are round and the size of grain is intermediate size. It is intermediate. The size is intermediate. The size is intermediate. So यहाँ पर आपके पास दो characters को control किया जा रहा है. There are two characters which are being controlled. One is shape. Second is size of granule. Size of granule. Now let's see again in terms of shape. देखिए जब BB है तो round seeds हैं. Shape is round. जब this small b है तो wrinkle seeds हैं. जब capital B है तो seeds are round. सो अगर हम शेप को कंसिडर करते हैं, सो दिस जीन इज फॉलोइंग डोमिनेंस, द फिनोमेना ऑफ डोमिनेंस, करेक्ट। सिर्फ अगर हम शेप को देखते हैं तो जब कैपिटल बी है तो भी राउंड है, जब कैपिटल बी स्मॉल बी है तो भी राउंड है, एंड कैपिटल बी स्मॉल बी इज रिंकल्ड. So if we talk about the shape, the phenomena of dominance is being followed, right? Now taking in terms of size of granule. Size of granule देखिए. In first case, the large granules are produced. In third case, small granules are produced. But in case of heterozygous, the intermediate granules are present. So in terms of shape of granule, this gene is following incomplete dominance. This gene is following incomplete dominance, right? So this gene is basically controlling two phenotypic effects. Mendel said only one phenotypic effect, but here it is controlling two phenotypic effects. That is why we call this phenomena as pleiotropy. That is why we call this phenomena as pleiotropy. Right, students? Well done. Now let's come to your homework for today. That is, first you will make notes of this topic. Then you will read these two topics that we have discussed. They are very small paragraphs in your NCERT. You will read them, you will make notes, right? And then you will send your work to me. Okay? And make sure you underline the important points in the NCERT. Okay, dear students? So all the best. Stay safe and continue your studies. Goodbye, everyone.